Hello, let's discuss about pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is multi-enzyme complex. It's kind of multi-enzyme complex that composes of main three components. The component one is pyruvate decarboxylase. We shall see the another component, which is the dihydrolipoyl transacetylase, and the last component is E3, which is dihydrolipoyl dehydrogenase. Pyruvate dehydrogenase complex is kind of enzyme that convert the pyruvate into acetyl-CoA pyruvate, which has been produced from the cytosol after glycolysis, to acetyl-CoA, which is introduced to the mitochondrial matrix for the Krebs cycle. Therefore, first of all, the pyruvate must be decarboxylated, whereby one molecule of carbon dioxide is given out. One molecule of carbon dioxide is given out. The enzyme concerned is pyruvate decarboxylase. This pyruvate decarboxylase has this component, thiamine pyrophosphate. Then the remaining these two carbon combine with the thiamine pyrophosphate to form hydroxyl ethyl thiamine pyrophosphate. Then the thiamine pyrophosphate will keep on rotating, will keep on recycling, while the so formed hydroxyl ethyl combine with the lipoic acids, the oxidized form of lipoic acid, to form acetyl lipoic acid, like this, under the component 2, which is E2, known as dihydrolipoyl transacetylase. Then after that, the component of enzyme 2, which is CoA, is going to be introduced to form acetyl-CoA. Under the enzyme dihydrolipoyl transacetylase. Then, after introduction of a CoA enzyme to form acetyl-CoA, then here we are going to form the reduced form of lipoic acid. Then the reduced form of lipoic acids, lipoic acid is going to be oxidized by FAD, flavin adenine dinucleotide, to form FADH, which is done under the component 3, which is dihydrolipoyl dehydrogenase, dehydrogenase, and which eventually NAD positive will be reduced to form NADH. So these are the three main components. First of all is E1, which pyruvate decarboxylase, play a great role for decarboxylating pyruvate to carbon dioxide. Then component 2, which dihydrolipoyl transacetylase, that introduces CoAs to form acetyl-CoA. Then the reduced lipoic acid then is oxidized. So the cycle is keeping on recycling. And the component 3, dihydrolipoid dehydrogenase, where fat reduces the reduced lipoic acid, then the reduced lipoic acid return to be oxidized lipoic acid. So at this component 3 is the point where oxidation and reduction and oxidation takes place. Apart from converting pyruvate to acetyl CoA without any intermediates, without forming any intermediates in between, also pyruvate dehydrogenase complex is regulated by two kind of enzyme bond on the surfaces of pyruvate dehydrogenase complex. These are the pyruvate kinase that inactivate by phosphorylating component E1, and another is what is phosphoprotein is phosphoprotein phosphatase that dephosphorylate pyruvate dehydrogenase complex to form active form of pyruvate dehydrogenase. Therefore, thank you viewers for listening. Make sure that you subscribe this video.